Good morning, everyone. I hope that you enjoyed last night, and by seeing some very tiny eyes, I suppose yes. So this morning we have the pleasure with Christian Bénard to chair a session dedicated to challenging setups for cutting-edge EEG and MEG research, and we'll listen to three talks that deal different aspects related to EEG and MEG research, from participant specificities to the hardware going through the analysis. During the first talk, we will see one of the most challenging but also exciting experimental setup consisting in working with young children. After a PhD in psychology completed at Free University of Berlin and a postdoc in the research group of early social development at Max Planck Institute for Human Cognitive and Brain Sciences in Leipzig, in Germany, our first speaker, Sarah Jessen, is now the group leader of Baby Lab at University of Lübeck in Germany. Her research focuses on neural bases of emotion processing, social behavior, and social perception in infancy and multisensory processing in social interactions. Today, she will give a talk entitled Recording and Analyzing Developmental EEG Data. Please, Sarah, the stage is yours.
Okay. Thank you very much for the kind introduction, and thank you first to the organizers for putting together this really exciting workshop, and especially for making it possible to have this hybrid version, because unfortunately I can't be there in person.
We can't listen to you. Can you unmute you, please?
Sorry?
No.
I'm muted? Can you hear me now?
No. Sarah, do you hear me?
I hear you.
You hear me, but we do not hear you in the room. So we have a technical problem here.
Anything from here?
It's not on your hands.
Someone from the staff, perhaps? Okay.
Sorry.
No, it's nice because in the meantime we can welcome more people so they don't miss any speak.
Can you try to say something, Sarah?
Hello, hello. Test, test.
But online they can hear Sarah. It's just us.
Sorry, just
yes.
Checking again, even though we checked a second ago. Where do I do that? Okay. I'm supposed to have, here it is.
Can you hear me now?
Sarah, can you try to say something?
Yes. Hello, hello, hello. Test, test. Can you hear me now?
This is the last day. We have less people in the room. We have much less volunteers. So normally we are like 3 doing that. I'm alone. and even the computers are tired, I think. Non? Non? Bah, la sortie de l'ordi. Ah ouais? Bah, à fond, ouais. C'est une bonne idée, ça. Ah, du coup, il m'a reviré. OK. I believe this one is going to work. Sarah.
Okay.
Whoa!
Can you hear me now? Yes? Perfect. All right, great. So let me start over again. So, all right. Welcome, everyone. Even on this last day, thank you for coming. And thanks especially to the organizers for putting together this super exciting workshop, and especially for making it available in a hybrid version, which at least for me has been working quite well for the past days, although now I'm even more sad not to be there in presence, because there were some really exciting talks over the past days, and I really would have loved to be there in person also. So I'm going to be the first one today in the session on challenging EEG-MEG setups, and as you already just heard, I'm going to be talking about analyzing developmental EEG data, and specifically infancy data. So my background is in developmental research. I do EEG with babies in social settings, and so I'm coming a bit more from the kind of applied side of cutting EEG methods, and I'm going to talk about how they can be applied in this special population in infants, and what you might want to consider, what's important to consider when dealing with babies and trying to implement these methods in developmental population. So, although I'm going to be talking about infants for my talk, I think, and probably most of you are not working with infants, I think a lot of what I'm saying might also be relevant to people working in other special populations, like patients, for instance, where you might have similar problems that you have with developmental EEG data. So I think a lot of the stuff I'm going to be saying can also be translated to other contexts, and might therefore also be relevant to people not working strictly speaking in infancy research, but also generally in research in areas where you don't have your normal typical student population. All right, what I'm going to be talking about, I'm going to start with some really practical aspects, just for a very few slides, what you would want to keep in mind when actually planning cognitive experiments with behaving, awake, participating infants and recording EEG, really from a practical perspective. And then for the main part of my talk, I'm going to be talking about MTRFs, encoding decoding models, and how they can be used in infant EEG research as an example of a more advanced EEG analysis method, and what you might want or would need to consider when applying them to infancy data. So I guess I don't have to motivate to this crowd here why it would be interesting to use EEG to analyze brain data, but I'd like to kind of highlight it from a developmental perspective, because in adults, we usually have the choice between a number of methods that we can apply, but in infancy, we are much more limited in the kind of methods that we can use to analyze neuroresponses. And EEG is still the most popular method when it comes to developmental cognitive neuroscience, especially in the really small kids, so in babies a year and younger, or also in toddlers. And this is the case for a number of reasons, which I think are pretty intuitive when we think about it. It's non-invasive, it's quiet, and doesn't constrain the infant. And this is like a main aspect why, for instance, fMRI is not possible in infancy research, and why kind of all the designs that we're using in developmental research are usually targeted around how can we implement them in EEG setup, what is possible to record with EEG in infants. So EEG is commonly used in infancy research, but of course it's still more challenging compared to when you look at a student population or an adult population generally. And I listed some of the challenges that we have when recording infant EEG data here. The first being that we have typically really short preparation times. Just to give you an idea, when we're working with adults, we have basically, well, not unlimited, but a long time to prepare for optimal impedances, for optimal signal quality. In infants, you usually want to be done in 5 minutes or maximum 10 minutes preparing your EEG recording, because you also usually want to have a happy infant participating in the experiment. And the longer you take to preparing the infant, the more moody and cranky it will get and won't participate in the experiment anymore. And then, of course, you can't instruct the infant to do anything. You can't instruct the infant to sit still, which is why you have usually a lot of artifacts in the EEG data that you record. You have motion artifacts, and the motion artifacts are not, as an adult, they might slightly move their head, but it's more like the infant pulling at the EEG cap or turning around. So you really have strong motion artifacts. And I will talk a bit about that when I talk about pre-processing later on, what the problem with those artifacts might also be, and especially in infancy data. And I think it's kind of intuitive to everyone why this is a problem generally for EEG recording. You also only have a limited amount of data because you can't record for an unlimited amount of time. So you're typically constrained, depending on your experiment, to 10, 15 minutes maximum. That's already super good for infancy research. So you don't have that much data and you have to work with the little data that you have. And I already mentioned that you can't instruct the infant to do anything. So you would want to design your experiment in such a way that the infant, although you can't instruct it to do anything, happily participates. You don't typically get any explicit behavioral feedback from the infant, besides maybe looking behavior. And you also can't instruct the infant to sit still, to only blink at certain times. So you also have to work with what the infant brings along. What are some of the things that you can do to still get good data from an infant when you actually do practical recording? Plan short recording times, that makes sense. But it also means that you, coming to the third point here, most infants won't complete the entire experiment. You typically want to design the experiment so that even if the infant doesn't want to participate anymore after 5 minutes, you still can use the data. And 5 minutes is really a realistic time. If you're talking about visual experiments, for auditory experiments, you can typically record for longer times because you can do all sorts of fun visual stuff along the side to keep the infant entertained while they are listening to stuff. Typically, you need a lot of participants because you have really high attrition rates. 50% attrition rate is pretty normal in infant studies. Of course, it depends on the specifics of the experimental design, but especially for visual experiments, you typically have really high dropout rates. But I think the more interesting point here is the last point, namely that you have to really put a lot of thought into what kind of stimuli you use for your experiment and how you can design the experiment and the stimuli to make them engaging for the infant to actually participate in the experiment. If you're doing visual stuff, you have to keep in mind the visual acuity of the infant, which is still developing over the first years of life. So the younger the infant gets, the worse their visual acuity actually is. So you want to make sure that they can actually see what you are presenting to them, that they can actually discriminate the fine details. And just as a rule of thumb, it takes about half a year, 3/4 of a year for the infant to be able to perceive faces the way we do at like a normal 1 meter distance. And as I said, you want to keep the experiment interesting, engaging to the infant. And so this all sounds like a lot of caveats to what you can actually do with experiments in infants. And now the question is, of course, given all these problems that we already have, why would you want to start applying cutting-edge EEG methods to that if you have already problems designing that kind of simple experiment? So as an adult, the classical, traditional way to analyze EEG data as infants has been to look at ERPs, especially in cognitive research. So there are a lot of studies looking at ERP research in infants. There are well-established ERP components that you can look at in infants that are super useful because they have been investigated in many studies, so we know a lot about them. But of course, there are certain drawbacks to using ERPs that are comparable to those that we have in adults that we talked about in the past days already. And as an adult and also in infants, there's a increasing interest in looking at brain responses to actual more naturalistic environments. So what happens if we don't have these super constrained experiments that are necessary to look at ERPs, but have more realistic input, have ongoing input, have continuous input? And we essentially want to look at the same research questions that we have in adults. I just highlighted two specific, maybe more specific infant questions here. One topic is language acquisition, of course, is a huge area of research where we look at how pre-verbal infants respond to language input, how variance in this language input affects the infant brain, how the infant responds to that, how different parameters influence the infant brain responses. And the other field that actually I'm more interested in is social learning, which is, of course, also a continuous, ever-changing stream of input the infant receives from their social environment. And these are questions, I think, that we can really benefit from using a more naturalistic design that is actually akin to what the infant is perceiving in their natural environment. And these are, so research questions are one reason why it'd be super interesting to use more advanced EEG methods that allow us for the use of continuous data, continuous stimulus input, sorry. But there's another more practical reason. If we use continuous stimulation, if you use ever-changing input, this allows us, from a developmental perspective, to design more exciting experiments. And I just mentioned that already. Designing exciting experiments is key to getting the infant involved, to actually getting good data quality. There are studies showing that if you use continuous EEG experiments as opposed to ERP, you typically have less data loss. So it's really a matter of getting good data that you design exciting, changing experiments. And of course, if you want to do that, you need a way to analyze the data. And this is basically why I think it's really important to look at these CuttingEEG methods beyond the question, but more from a practical perspective as well. And the remainder of my talk, I will discuss the question using encoding, decoding models as an example. Can we use them for analyzing infant EEG data? What do you want to consider when doing that? So I will be talking about MTRFs. And we had this really great talk from Ed Layler on the first day. So I think you all know basically the method I'm talking about. So I'm just going to focus on the infant-specific aspects that are relevant in this context. And use one experiment that we did with 7-month-old infants as an example to just demonstrate what is feasible in infants and how this compares to responses in adults. So these are basically the topics I'm going to address. I'm going to mention some aspects to consider in pre-processing. Then I'm talking about generic versus individual models, about the temporal unfolding, where we find the best, basically predictive accuracy, and the comparison between AND and decoding models when looking at infant EEG data. So the study I'm going to be talking about, what we did is we had 7-month-old infants, 52 infants. And what we did is we just showed them a 5-minute long cartoon of a Peppa Pig. If you don't know Peppa Pig, it's this child cartoon series of a family of pigs going about their daily lives and having some adventures. And we just used the video clips as they are, didn't do any manipulation on them, and had the infants watch the video clip. Excuse me. And for comparison, we also had an adult group of 26 adults who also watched the same video clip. And then what we want to know is it possible to, from this continuous stream, extract neurotracking responses in the infant comparable to what we can do in adults? And we were interested in both auditory and visual processing. So we focus in this experiment on 3 input parameters. We extracted the auditory envelope. We looked at the motion content, and we used a pretty crude way to quantify motion here. So we just used the change from one frame to the next as the motion vector. There are better ways to do that. We can talk about that later if you're interested. But for now, we just use the change in pixel from one frame to the next. And as the third regressor, we used luminance. So change in luminance from one frame to the next. As I said, it was like about 5-minute cartoon. And as an inclusion criterion, the infants had to contribute or had to have at least 100 seconds of usable EEG data. And as I mentioned in the beginning of my talk, typically in infants you have pretty high attrition rates. So we tested an additional 26 infants, but couldn't use them because their data quality wasn't good enough, or they didn't even watch the entire movie. I said in the beginning, 50% attrition rate is common in infant research. We had a much lower attrition rate here, which in my opinion also speaks to this being actually an experimental design that's kind of interesting and appealing to infants, and they like to participate in that here. Pre-processing, there are some aspects that I think pretty standard. So we use an average reference. We use the filter from 1 to 10 hertz, ICA. And then there's always the question in infants, even though we have infants that are happily participating, there are typically some stretches of data where the data quality is not so good, so we can't use that particular stretch of data. So there will be epochs that have low data quality and that we did not want to include in our analysis. So what we did is we segmented the entire continuous stream in 1 second epochs and then rejected those epochs where the data quality was bad and replaced them by zeros. I'm going to talk a bit more in detail about these two aspects using an ICA for pre-processing and this zero replacement, because these are the two aspects that we often get questions to. The first one is the ICA, which if you are working with adults, it's probably for most a pretty common step in pre-processing. For infancy research, it's actually not so common to use an ICA as part of your pre-processing for several reasons. So the first is that what an ICA, so the idea behind an ICA is that you can detect stereotypical repeating artifacts that reflect in one or very few ICA components and that you can then reject. And this is often not so, this approach is often not so successful in infants. One main thing that you typically use an ICA for in adults is to remove components of the signal that are linked to eye movements, eye artifacts. One thing here is infants just don't blink as much as adults do. So I brought you some data here, some numbers for that here. If we look at really young infants, they blink 0.7 times per minute on average, and this increases a lot as people get older. And by 20 years, which is the typical participant age, you have a blink rate of 4 to 17 blinks per minute. So on the one hand, eye blinks are not as much a problem in infancy research as they are in adult research, because babies just don't blink so much. On the other hand, you can't instruct the babies not to blink at certain times, so you will have blink artifacts in there, but they typically have less stereotypical typical topography, so they are really a lot more difficult to extract, and they are also harder to separate from the EEG signal that we actually want to look at, because overall the EEG responses in babies are slower compared to adults. So this is generally true for the EEG responses. Also, if we look at oscillations, for instance, you can see what we typically observe in adults as alpha frequency range, for instance, so like 8 to 12 hertz, is in infants more like, depending on the age, 6 to 9 hertz. So infants are generally, everything's a bit slower in the EEG, but it's also, that's one component that contributes to it being harder to use ICA to discern these stereotypical artifacts from the signal. So in some, ICA as part of a pre-processing, I think is not always necessary. It might be in some cases where you have very maybe strong artifacts, where you also might maybe have very stereotypical artifacts for some reason related to the experiment. But in many cases, it doesn't provide a great benefit to pre-processing. So I would see this step really as optional. The other aspect is the zero replacement I was talking about. So we have stretches of data that we need to remove. It doesn't make sense to replace them by zeros or not. And the question is always, okay, if we replace too much data with zeros, what does it do to our analysis? And we looked into that a bit, and looked what it does to the temporal response function, which I will talk about in a second, if we replace more and more data with zeros due to this just noisy data being there. And you can see that if we replace, so in the original data set, we replaced 15% of the data because they were noisy. If we increase that to 25, 35%, it doesn't change so much. it kind of flips when we get like 45% or higher. So there's quite a high amount of zero replacement that can be tolerated in the signal, but of course it still raises the question, how good is the data quality we have, and how good is the remaining data that we have? Because if we have a data set where the quality is so bad that large stretches of data need to be replaced, chances are high the remaining data set is also not of such a high data quality. So in general, in our experience, the zero replacement is a pretty good way to deal with these interruptions in the continuous recording that we have, and allows us to keep infants in our sample that we would otherwise have to exclude. Okay, so that's all I wanted to say about pre-processing. What do we end up in the infant data when we do these pre-processing steps? This is kind of the MTRFs that we get out of the data set. And I'm going to talk you through the picture that I'm showing you here. I think you have seen MTRFs in adult research over the past days. And what we get in our data set here with the cartoon movie is what you see for the adults here in blue. In A, you see the auditory response function. In B, you see the response function to visual regressor, in particular the motion regressor. And in red, you see the infant response. No, sorry. In blue, you have the infant. In red, you have the adults. Sorry if I'm mixing that up. So the most important thing that I would like to highlight here is that we are able to get a distinct, clearly defined response function for both the auditory and the motion regressor for both infants and adults. Directly comparing infant and adult, and they are of similar magnitude in infants and adults. The pattern looks slightly different in infants and adults. It's always kind of challenging to directly compare brain responses between infants and adults. So this should be taken with some caution. But what you can, for instance, see in the auditory response function is that for the adults, you have this pretty prototypical 2 peaks there around 240 milliseconds. For the infants, you have only this 1 peak, which is peaking at a similar delay as the adults do. In the motion response function, you kind of see a more similar pattern in the infants and adults. So they both have these 2 peaks. Sorry, you don't see the pointer, right? There's 2 peaks there, but the infant pattern in blue seems to be slightly delayed, which I think fits nicely what I was saying earlier, that generally in infants we have slower brain responses. In the bottom, what you see there are significant differences between the infant and adult data, and you can see that there are stretches where there are different, but in general that we get response function in both cases. So the first question I want to talk about is individual versus generic model. And as you are probably all aware of, the general concept between computing these temporal response functions is that you train the model on a subset of data and then use kind of a left out sample of the data for testing the model that you train. And we tried 2 different ways on these infant data to compute the temporal response function. We tried to use an individual model and a generic model. Using an individual model is probably the more common approach in adult research. So you take the data set of a given participant, split it into, 80-20%, use the 80% for training, and then the remaining 20% for testing the model. And this works, I think, pretty well if you have enough data for a given participant. We were kind of worried that we might not have enough good data from the infants, so we tried a different approach, namely using a generic model where we did not split the data of an individual infant, but actually use the entire data set of n-1 participants to train the model to predict the response of the nth participant. And when we compare these different approaches in the infant data set, you see here again the audio and motion response that you saw on the previous slide. On the very left is the pattern, the predictive accuracy we get when we use the generic model. In the middle, you see the individual model. On the right, you see just a shifted control condition where we don't expect to be any predictive accuracy. What you can see here is that we get overall comparable responses using both the generic and the individual model. So both approaches seem to work in the infant population. We have here, especially for the motion regressor, slightly better predictive accuracy for the generic compared to the individual model. Especially we have a higher variance here for the individual model. If we take both regressors, the audio and the visual regressor in the model, you get a slightly better predictive accuracy for both the generic and the individual model. But you still have the same pattern that the generic model seems to achieve a slightly better accuracy compared to the individual model. Looking into that a bit more detail, we can see that what you see here, sorry, mine doesn't working. What you can see at the top right with the purple dots, we have the correlation between the individual and the generic model, and you see that there is a pretty good correlation between the two. So both approaches seem to be going into the same direction. What you see below that is the number of infants where you have a better performance for the generic over the individual model and vice versa. And again, you can see that for most infants, the generic model seems to do a better job compared to the individual model. When we compare that to the pattern that we see in adults, so comparing generic model versus using an individual model for infants and adults, you can see first that the generic model also seems to perform slightly better for the adults. So in the left panel A, so in red you have the adults, in blue you have the infants. In the left panel you have the generic model, and you can see that for both infants and adults we have a slightly higher predictive accuracy for the generic model. What you can also see, by the way, is that we have in both models a better predictive accuracy for the adults compared to the infants, but that for both models we are able to generate a prediction for the infants as well. So at least in this experimental setup, for these data, the generic model seems to be doing a better job here. Then the second question that we ask is, okay, what time range are we actually looking at? Because for the data that I was showing here, we used a range of time lags between -200 and 800 milliseconds, so like 1 second time range around where the temporal lags could occur. But maybe this is not the optimal time window to actually look at the predictive accuracy for the infants or for the adults. And so what we did is for the entire time range where we looked at the temporal lags, we computed the predictive accuracy and then looked at the time windows, which are marked here in orange, where we observed the highest predictive accuracy and checked whether we actually get a better performance if we just use this time window as opposed to the entire time range of temporal lags. On the left, the infants, on the right, the adults. And what you see is that for the infants, it does not seem to help if we reduce the window where we are looking at the predictive accuracy, because you can see that we have a higher predictive accuracy if we use the entire time window here on the left as opposed to the optimized time window that you're seeing here. For the adults, we can have the reverse pattern. There, it actually does help to use the optimized time window. We get a higher predictive accuracy if we just use this window of 150 or 100 to 400 milliseconds time lags. And if we look again at the on-to response function, this is kind of corresponding also to where we see the peak in the on-to response function. So for the infants, it does not necessarily, it might be specific to this experiment, of course, but in our experience, it doesn't necessarily help so much to narrow down the time window we are looking at, but it might help more to use the entire time window of temporal lags. Then kind of one thing I haven't talked about at all in this experiment so far is the luminance regressor. So I said in the very beginning, we used motion, we used the auditory envelope, and we used luminance. thing is, for luminance, we did not get any reliable response function. So here on the left, you see the infant participants. On the right, you see the adult participants, and you can see it's basically a flat line in both cases. Also, if we look at the predictive accuracy, it's basically 0. So not everything seems to work so well in this experiment. The reason why we think we were not able to get any reliable model for the changes in luminance is that we just had very little variance in luminance over the experiment. So as I said in the beginning, we did not manipulate the video in any way. So it's a children's cartoon movie and there just isn't much change in luminance. I would expect that if we specifically manipulate luminance or use some material where you have naturally higher variance in luminance, that you should, in theory, also be able to get a response function to changes in luminance. But with the stimulus material that we were using, this was just not the case. So, to sum up what I have been saying so far, I think with this experiment we could demonstrate that we can use MTRFs successfully in infants, as can be used in adults. Different regressors seem to work differently well. Generally, the more variance we have in the regressor, the better results you probably get, or the other way around. If you have too little variance, it's not possible. The joint model yields a slightly better performance compared to using only the audio or only the motion regressor. And based on the data that I have presented to you so far, the generic model seems to work slightly better compared to the individual model. The last thing I want to talk about is this last point here, generic versus individual model, because we looked at a different data set to look a bit more into this point. And in this different data set, different study now, but again, 7-month-old babies, we had 7-month-old babies listening to their mother reading a children's story to them. So this time it was a purely auditory experiment, and the infants were listening to a children's story being read to them. And we again looked at the predictive accuracy, so at the neural tracking strength we call it here, for the individual versus the generic model. And here you can see, what you see here is that we have a slightly better performance for the individual as opposed to the generic model. So here we actually see the reverse pattern of what I was talking about in the first experiment today in my talk. What's the difference here? There are actually several differences between the experiment, so we can't really say what's the reason for that for sure. The first is that in the initial experiment with the Peppa Pig video, we had 52 infants. In this data set that I'm analyzing here, we only had 10 infants. And of course, it makes sense if you compute a generic model based on other participants. The more participants you have, the better your model performance will be. And so if you have too little participants, using a generic model might just not work. So you need enough participants to be able to do that. On the other hand, we have slightly longer data here. So here it was a purely auditory experiment, so the infants listened a bit longer. We had an average 7 minutes of data as opposed to 5 minutes in the original experiment, which again works in favor of the individual model. So we have more data from an individual that we can use for the model computation. But I think the most interesting point here is that we get a strikingly different pattern if we look at decoding as opposed to encoding. So in the first experiment, we only basically used encoding models. Here we actually went also the other way around and looked at decoding. And you can see that here you have the model performance for the individual model clearly outperforms the generic model. So depending on which type of model you use, it might be advisable to use the individual as opposed to the generic model when looking at the EEG data in the infants. So to kind of extend the summary I showed you in the previous slide, generic or individual model also seems to be a question of the general experimental setup, how many participants you have, how much data you have available, and what sort of model you want to use. Okay, this is basically the end of my talk. So just a quick overview over the kind of experience we made with using MTRFs in infancy data. If you're interested in this type of research, I brought you some further literature that's basically going into more details on that. So the first two are just papers looking at problems or solutions for EEG recording in infants. The first one is specific to ERPs, but I think a lot of points also hold for other EEG research. And the second one is actually dealing with more advanced EEG methods. Then we have a manuscript, which is basically the basis of this talk, that's on PsyArchives. And generally, I'd just like to advertise this upcoming special issue in Developmental Cognitive Neuroscience that's specifically dealing with different cutting-edge EEG methods for developmental cognitive neuroscience, and is providing, I think, a really nice collection of tutorial approaches and how they can be implemented in developmental EEG data. Yeah, and with that, I'd like to conclude my talk. Thank you all for your attention. And again, thanks to the organizers. And thanks to all the families for participating in our research. Thank you very much.
Thank you very much, Sarah. Do you have any question from the audience? Yes. One.
Yeah, first, thank you for the very interesting talk. So I was wondering, why do you think the generic model performs better than the subject-specific model? Because I think it benefits from having more data available, but at the core is really that there are differences between individual subjects, right? And so I was wondering, have you looked into this also from like the feature or the EEG signal itself? So how does that differ between subjects? And also, if you have looked at any transfer learning techniques, for example, that you have a generic model which you then try to adapt with a new subject.
So you mean whether we looked at the kind of how much variance we have between individuals when we try to compute the generic model?
Yeah, so because I think what you basically did is just look at the decoding performance in the end and not really at the signal level or at the feature level, which you try to use to, or which you use to train the model.
Yes, yeah, that's of course a very good point. So it might be a prerequisite for successfully using a generic model is of course that we assume that all infants respond in a similar way to the stimulus. And I think in our case, this is generally a reasonable assumption because what we had was just a focus on pretty low-level sensory processing, so just auditory input, visual input. But I think especially if you move into the direction of a setup where you might expect stronger individual differences. So how much is an infant paying attention to this or that, or maybe even in a learning context, or also in developmental context, like which infants are maybe more developmentally advanced compared to others. This is, of course, a huge issue. We haven't looked specifically in our data set into individual difference in response to the individual input parameters now. So we just looked at the performance in the end.
Any other burning question?
Yeah, it's not really a burning question. I just have a small technical question. So what did you use to do your pre-processing? Because you're dealing with very noisy data. And it looks like you did your topo maps with MNE. So did you use MNE for pre-processing?
We used field trip and and just built in MATLAB, just MATLAB functions for filtering.
Okay, because in MNE there's a very smart artifact rejection correction tool that does not use any fixed thresholds to reject a trial, but it computes the rejection threshold based on.
Sorry, I can't hear you anymore. Hello? It seems like I might have lost connection.
I think you're back.
I'm back. Okay,
Yes.
great.
Did you hear the question?
I heard the question, and I heard the first part of your point. I think saying that the M&E toolbox has this really interesting sounding approach.
Yeah. It's just a little, it's more like a comment than a question.
Yeah.
I think that could just help you to save a lot of data because it doesn't reject a trial on a predefined, a priori defined threshold rejection, threshold criterion, but it computes the criterion in a data-driven way, and it can help to save a lot of data.
Okay, thank you.
You might want to try it.
I'm definitely going to look into that.
Yeah,
Thank you.
It's called auto reject, and it's in
Okay.
M&E. Okay.
Thank you very much.
You're welcome.
I think we can thank again Sarah for her presentation.